Okay, my friends, this is Diana Cohen. She's called Physics Girl. Very nice. Goes in, she's an MIT physicist and all kinds of stuff. She's desperately ill at the moment with long COVID. But I went to search on dark matter, and this popped up very briefly. And she's saying what makes up 95% of the universe is dark matter and dark energy. Well, I decided I would to see how she's doing because I knew she was desperately ill. And apparently she's not getting better. Now, this is going over a year and a half. So I have a couple of statements to make about that. I give no medical advice whatsoever, but I am a researcher, so I will present some research just been done that just in the interest of research. I leave it from there. I don't make any recommendations, do this, do that. No recommendations whatsoever. But what is the state of the current research on long COVID? Let's take a look at that, and then we're going to do the dark matter too. All right, we're going to get to dark matter. Dark energy makes up 95% of everything, as Diana Cohen has to say. Now, this is tragic. She says it's impossible not to cry every day. The disease has taken everything. For a year and a half, I've lived in a nightmare, made no progress getting out. Severe long COVID is something no one should have to endure. And she's in desperate straits here. And I mean, it's life-threatening and it's absolutely life-altering. Now, what does the research say about this? Again, I make no recommendations whatsoever. Let's see what the research is saying. Okay, my friends, this is kind of a strange twist today. I was getting ready to do the electron flood theory and dark matter and dark energy and what is the universe made of. So I click it, and it pops up with Physics Girl, she's fabulous. And she says that 95% of the universe is made of dark matter and dark energy. So I'm thinking, this is good. I used some of that. And then I thought, I know she was sick with COVID, and she had long COVID. So I decided, well, I'll go up there and see how she's doing. It's not good. And I have some information and research on COVID. I'm just going to put it out there. I make no recommendations whatsoever. I do research. Without research, there's nothing. Everybody's got to be quiet and sit in a corner? No. You can show what the professionals are doing. This is not what I'm doing. It's not my research. I'm saying here's what they're showing. And let's think about it, that's all. So don't think I'm giving any medical research, I'm giving medical research. I am not giving medical advice. No medical advice here whatsoever, all right? Please, so don't have Facebook or, YouTube or anybody stop me from this, because we need to look at the information. This is strictly information-based. Okay, it's happened again. I cannot do something partially. It's just my nature. When I started looking into this, I'm not going to be able to do the dark matter inside of this video because let's talk about the tight junctions and about what your immune system is all about. What does the research indicate? Because I'm telling you right now, it looks to me like invasion of these viruses and flu and all that stuff comes through tight junctions. If you have tight junctions, you're not going to get invaded. And that's exactly what they say. Tight junctions. This is inside your lungs. So you have tight junctions, adherent junctions, desmonas are all types of cell connections. They keep the cells together. They connect cells together. So they're glued together, literally glue, and it's a slurpy glue. And it actually is called slurpees, small leucine-rich proteins. They're gummy and gooey, and you can move and stretch and glue and all, but nothing gets through. No. So let's talk about tight junctions. These junctions are located in the plasma, that's the fluidy membrane between cells. So the cells come and they bump together, and there's a plasma fluidy membrane in there. It creates a watertight seal. Tight. They perform two important functions. Now listen. They limit molecules and ions, so you can't. Nothing coming through there. You stay outside. Everything else goes. Your lungs go that side. Now, so they limit molecules and ions. The tight junctions prevent molecules and ions from passing through the space between the cells. Your lung cells and so forth, that's a whole different story. They have their own mechanism for allowing things out. Now, they block membrane proteins. Membrane proteins, they also stop integral membrane proteins from moving between the apical bumps and basolateral surfaces of the cell. We'll look up that in a second. Tight junctions are essential for maintaining the integrity of epithelial and endothelial barriers, which is important for homeostasis. In other words, if you don't have the tight junctions stuck together, glued together, you're going to get invaded. It's really basically as simple as that. Okay, so what do we got? Tight junctions are inside of these gooey things that stretch and move around, which is inside your lungs. And the tight junctions, adherens, and desmons are all types of cell junctions connect cells together. Well, the tight ones are the ones we're talking about now, because primarily those are in your lungs where you're getting infected easily. Now, these junctions are located in a plasma membrane, that's the fluid membrane between cells, and create a watertight seal. So the cells are bumped up to each other, and nothing can get through. They perform two important functions. They limit molecules and ions. Tight junctions prevent molecules and ions from passing through the space between the cells, so they can't get in to invade you. They block membrane proteins. Now, don't forget proteins. They block membrane proteins. They stop integral membrane proteins from moving between the tip of the apical part out here to the basement layer, which is the basolateral surfaces of the cell. So they kill the stuff before it gets down to the cell surface. Tight junctions are essential for maintaining the integrity and epithelial and endothelial barriers, which is important for homeostasis or stability of your body. So remember this. They're talking about proteins. These are where it's integral membrane proteins. That's where they're killing the stuff from the top of the cell, from there to the surface. So they never get down to the surface. And they can't get around the cells to invade you. So the cell surfaces. Let's look at the cell surface of a membrane. All right, this is the membranes. Now, all membranes basically are bilipids. It's a phospholipid bilayer. So this is more phosphorus, and in between there's hydrophilic and hydrophobic things in here. One repels and one attracts. There's nothing you really have to know about. Just remember this. There's a lot of fluids in here. And out here is that apical that I'm talking about, the outside bump, where all these proteins are. Proteins are just like enzymes, and they sit on the outside bumps all over here, and all of these bumps are glued together. You can't get in between these bumps. They're glued together with like a slurpy membrane around them, and they glue and they move around like this and that, but they don't come apart. There's no penetration allowed. And if it does happen, there's a fluid-filled highway in here, which is called the interstitium now. This has been recently announced, that will attack that instantaneously. And here it is right here. You see this? This is what they normally consider was a membrane. Well, there's more to it than that. There's another layer which they always considered was, I guess they thought it was just part of this as a flat plate, because by the time they autopsied, there was no fluid left in it. So this is meet your interstitium, a newfound organ. This is 2018. They didn't realize this even existed. And this is like your skin. There's the fluid-filled highway. Now, by the time they had an autopsy, they were flat. There was no fluid, so they just thought it was a flat sheet. And underneath is the membranes and so forth, and then you get into your tissue and your interstitium, the things that stretch. And anyway, this is where all of this immunity comes from. You can get invaded so much, but when you get through there, now you got a problem. But you got a fluid-filled highway. You got so much fluid in you. There's 4 times more lymph fluids, which contain these enzymes and so forth, than there is even blood in you. So you should easily be able to come and overcome whatever invades you if you have the correct enzymes. And to have the correct enzymes, it appears that you need the correct bacteria. Let's look into what bacteria do to create the enzymes, and what the enzymes do to create the chemistry to attack things. Because enzymes do what they call click chemistry. I call it anyway. And when an enzyme attacks something, it pops open like this and it goes click, 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 and it kills them just one right after the other. And it doesn't even get used up. It just stays the same thing and wipes out scads of invaders, or breaks down fats and proteins and carbohydrates, sugars and all that, because they're the same sort of thing. The same thing. It's just a chemical, little chemistry set. It comes up, goes pick, 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 and they're very, very exotic, because every one of these little attachments here, you see this? Every one of these little blue and red looking things is what they call a magnetic moment, a dipole moment. All right, and you have to have exact signature. How elegant that is unbelievable. And when it comes up and it sees something that fits this signature, it goes click, 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 you're all dead. But it does it so like done. And if it wants to build something, the enzymes that pop out of here are so destructive to molecules, to their half-lives, that it goes and it just breaks it down, and then it reassembles it to whatever it wants. And that's why you can take a element here and invade it with an enzyme, which is this, and it will make that element go down or up. I can make copper go up to zinc. Decay doesn't mean it goes down. It can go up or down. That's what they don't understand. They say from iron, they can't understand how all these elements formed up here. They just don't seem to fit what they consider for their standard model, because that doesn't work anyway. But when iron gets attacked by enzymes, it can go up to cobalt, it can go up to nickel. I think it can go both ways. They all go both ways. They all go both ways. You can add extra, because it breaks it down and puts it back together. It can put it back together bigger or smaller. That's just the way it works. All right, you see this? These are isotopes of copper. And they decay and they change. Isotopes means they're not exactly the same as a full-size copper. A copper, there'll be an area where it's stable, and then there's going to be instability. Now, that's radioactive decay is the process by which an unstable atomic nucleus, that's the core, loses energy by radiation. Well, what is it radiating? A material containing unstable nuclei is considered radioactive. Well, what's it radiating out? It says 3 of the most common types of decay are alpha, beta, and gamma. All right, well, you see what happens? Little bits and pieces are popping out of there. That shouldn't be. If it's supposed to be only protons and neutrons, we're losing bits and pieces of the protons or little bits and pieces of the neutrons, which you are. In the standard model, it does not account for this. It doesn't work. The standard model doesn't work. I mean, I've been showing this for a lot. Now, the reason it doesn't work is because instead of these being protons and neutrons like this, and little bits and pieces falling off that they shouldn't, each one of these is like this. And then, yes, little bits and pieces can fall off, no question whatsoever. And more can get attached here than they should. So what do you end up with? You end up with one zone in the middle that's stable, but sometimes they don't have enough, and they want more, and sometimes they have too many and want to give up some. That's called a decay. All right? So here we got copper. Copper is above iron, which it shouldn't They don't even know how all this stuff happened. Copper is very interesting because that's the one that really conducts electricity. But anyway, there's two stable isotopes. Isotopes means it's just not the full size. So here we got, let's go right up to stable. All right, they got one stable up here, and then they got a stable up above. So this is like a plus spin stable and a minus spin stable or something like that. Now, every one of these is copper, but they're in bigger sizes. You see this? Just 29 and 26. All right. 55, 56, 57, 58. This is all the mass. The mass, the bigger the mass. How big is it? Well, how could it be all these different masses? And every time they add a neutron, in my world, that's adding almost 2,000 particles. So instead of 54 particles, which is 29 protons and 26 neutrons, that would be 55 times just about 2,000. So this is not 55, it's 2,000 times 55. And all of these are 2,000 times whatever that number is. All right, so these are all different coppers. Now look what can happen to copper when they get the daughter isotope. A daughter isotope just means it's after the decay mode, where however it decays, you end up with nickel. So copper can go down to nickel. That's what they consider that the decay. Okay, it's copper down to nickel, that's decay, that's the end of it. No, it's not. Because it can go up. It can also even go down further. It can go down to cobalt. Nickel, nickel, nickel. It can go up to zinc. Copper can go up to zinc. That's way down here. It can go up to zinc. Where is it? Right here. Right between the stable states. It's drifting between nickel and zinc, between the stable states. And then up here, it's all zinc. Well, there's some copper. It decays down to still staying copper, but it's what they're saying. Well, look at all these. These are all different states of copper. Can you imagine that? And they decay up to zinc. That's because of a nucleosynthesis, which means here's the nucleus, and it is at a certain size. All right, and then along comes an enzyme and goes, and it puts it back together at that big. Those go somewhere else, do whatever the enzyme wants with them, takes them, moves them, whatever. Or it may bring some other guys over and add on to it. That's what's happening here. The zinc is getting bigger than the copper. So that's the whole idea with nucleosynthesis, and that's not understood. I don't think that's understood at all, to be honest with you. Nucleophilic substitution is what I studied deeply because it works with my mud fossils. But nucleosynthesis is what it actually boils down to, is the end result is the nucleuses end up getting rearranged because of the enzymes that are in there. Because everything that ever was alive is saturated with enzymes. That's why carbon-14 dating doesn't work. When they take something that was ever alive and they try to date it with carbon-14, it's just not going to work because the carbon-14 has been already affected by enzymes. So it's never ever going to work. See, here it is right here. Enzymes have a short half-life, and they just go brrr, and that's what cascades down and breaks down proteins and other things. Listen to this. Enzymes' short half-lives are important for maintaining their optimal activity, the activity of continuing to break down stuff, which is the protein turnover and degradation, breaking it down, the ability to adapt to different cellular conditions and needs. As the things break down, they have to change them again and then reconstruct them into whatever the cell needs. It allows enzymes to respond quickly to changes in the cellular environment of metabolic demands. As it's being chewed up, and metabolic means like you're eating it. As the cellular environment or metabolic demands, it can change rapidly. which ensures the efficient functioning of metabolic processes and overall cellular homeostasis, which means you're good to go. The cells are going to be working fine. But you have to have these enzymes, and the enzymes have half-lives, and I don't know if they understand the reason behind the half-lives is because they're breaking down all these other molecules. It sounds like they do. You know, protein turnover and degradation. That's why they have to have these short half-lives. So I guess they do understand it. So apparently they understand that the bacteria are the factories that produce the proteins. Bacteria can produce proteins through a process called bacterial protein synthesis. Bacteria is just amazing stuff. Proteins, large, complex molecules, long chains of amino acids, like these things here. They call them polypeptide chains. And they come out and they fit into the ribosome ball, and they turn into just a nice little tiny round ball. And then they run around through your body, and these proteins are used to build. Some of them are building blocks. They do all kinds of things. Proteins are basically the same as enzymes, but I think they have like an extra range that makes them into building blocks, as well as like a middle range where they sort of add and subtract, and then the enzymes go all the way down to where they're just complete destroyers. Like, everything goes. And they probably little adjusters, and then they get, little ticklers, and then they get to the other end where you're up in the protein construction bits. So it's like a spectrum. That's the way I'm looking at it right now. And there's so many bacteria, you can have that full spectrum, no problem whatsoever. All right, so this is fully understood that bacteria create proteins. All right, so that's bacteria protein synthesis. And they are used to reproduce, repair, and undergo basic metabolic processes. They chew up the food inside of you and make it get ready for you to be able to digest into those little molecules. They reconstruct the molecules using enzymes. These shoot out the enzymes. See, they're all the same. These are the ribosomes, little balls. And I show this pretty carefully here. That these are where the ribosomes come out. And they can either turn into enzymes or proteins. Is there really much of a difference? Like I say, I think there's a range between the total destruction choppers of enzymes down to where they're little manipulators, all the way up to where they don't really break anything. They start to add a little bits and pieces up to where they can replace bones and muscles and everything. So it's a spectrum. That's the way I see it. All right, let's go back to these junctions again. This is the most important thing for you to take away is every one of these proteins, every one of these different enzymes requires a different bacteria. That bacteria has to live and be functioning and living in your body to create the ribosomes, which create the enzymes when they're needed. So when you have tight junctions, you need all of those different types of enzymes and bacteria to create the gooey stuff. All right, so let's just think about again what these things do. They isolate you from everything from penetrating down. All right, so these are the different junctions. The adheron junctions are made of cadherins, which are transmembrane proteins. Once more, membrane proteins. It forms a continuous adhesion belt around each cell completely. They provide strong mechanical attachments between cells and are closely associated with actin cytoskeletons. They're really tough. Now, then you even get tougher, I believe, is even more tough. The desmones. These junctions are even stronger than Heron's junctions and connect intermediate filaments from cell to cell, and they're made of desmona intermediate filaments. I'd say they work out basically like this, and are a network of cadherin proteins. Again, proteins made by enzymes, the same thing, enzymes, proteins made by bacteria. and intermediate filaments. The desmonas also contain intercellular anchor proteins, again proteins, called placoglobin and desmoplankton. Those are the anchors. All right, so because once they pull apart, you're done. This anchor's got to be good. That requires a certain type of chemistry. The filaments have to be good. Another chemistry. It's chemistry on steroids, and it has to be done instantly, and that requires enzymes. Okay, now remember, these are the cell junctions. These are the tight junctions up here. They're supposed to pinch tighter so nothing can get through there. Then you got your adheron junctions. They adhere, they hold on, and they can do this kind of stuff. And then you got these other ones they call desmonosomes, which are these down here. They're something similar to this. But every one of these is layers in the tissues. Now, then you've got gap junctions, and you've got these basal lamina down here, and the hermodesmonas there's all kinds of things going on in here, and that's just in one cell, or cell membranes. All right, these are different kind of cell junctions, and they hold the cells together. If you don't, that stuff gets through down into this basal lamina. Basal means your basement area, and then you're done. It's whoops. All right, just for clarification, these aren't completely different cells, these different cell types. Their tight junctions are within the same cell, and they're right at the top. They're towards the apical area at the top, and they contain, in this area up in here, is going to contain all of those fluids they're talking about, the enzymes. Below that, you have the adherent junction, which is just like a layer of the basement, basically, below this plasma membrane area where that's that apical part I talked about. Then below that you have these desmones that are another junction layer that holds them together. And then you got a whole bunch of other things going on in there. And then at the bottom you're through your membrane, and you're into your good tissue that you do not want to be into. So you want to keep all that stuff out. And the first line of defense is this one here. That's the apical area. All right, and in this fluid-filled area here is where all those enzymes exist. And I believe these little dots going around here are basically lymph ducts, because this is the fluid-filled highways up in this area. All right, just be sure you understand, the bacteria create the proteins. If you don't have the bacteria, you're not going to have the protection you need to make these different fluids and so forth. They all have different chemistry, every single one of them. That's why you have so many of these ribosomes floating through your body. You need some chemistry. We got it, no problem. No bacteria, no chemistry. All right, it's simple as this. There's a gigantic paper on this. In summary, the airway epithelium is a dynamic barrier. That's the layer that the air is supposed to go past, and nothing's supposed to come into you. It's a dynamic barrier, constantly responds to luminal stimuli and coordinates its response to maintain homeostasis in the lungs. So you need all kinds of ribosomes ready at the demand for the kind of conditions you're in at that time. All right, breakdown in this coordinated response can lead to a myriad of lung diseases. A myriad means bazillions. All right, and they know they're into this pretty good now. They understand it, that it's somewhere up in here. It goes on and on and on and on and on and on and on. It was way up here somewhere. I had something highlighted. Here it is. Intercellular junctions form adhesive forces. Adhesive, they glue it together. Connect neighboring cells and separate the external environment from the environment below, which is your good tissue. The three main types of these are the tight one right at the top that has the enzymes in it, and then the other one right below that seals that membrane layer up above with all the fluids in it, and then the desmonas is down below that seals that from this layer. All right, and the tight junctions are apically located, so they're at the top, the tight junctions with all of their fluids, and they have all of those multi-protein complexes consisting there in the transmembrane area, which I showed you. So it seems pretty obvious to me that this is where, you know, the area of penetration. Now, how do you fix that? You need to fix your bacteria, basically, because that's, and they seem to understand that now. That appears to be the case. Now, don't forget, bacteria are your enzymes, and they're your digestive system, your digestive enzymes, and they're all the enzymes. They're basically created by bacteria. So you have to have a very thriving environment of bacteria living in you, of the good stuff. You don't want the bad stuff, but you do need some of it, because you need stuff to clean you out. So you need actually flesh-eating bacteria inside your body to eat the stuff that dies. You're dying all the time. So there's all kinds of things going on in you, but it's kept in check by a balance of good and bad bacteria. But once you lose the good bacteria, the bad take over. Once you lose the bad bacteria, the good, sometimes they pile up. The bad bacteria isn't there to clean stuff up. The bad bacteria sometimes do the jobs the good bacteria refuse to do. All right, so once again, we know it's all related to the endocrine system, and it's all the gut-brain axis, and then you got the lung gut axis. All it is we're making enzymes for you up there in the lungs. I know what you use up there. You need this gooey stuff and that kind of gooey stuff. We'll give you some enzymes to do it. Oh, you over there in the brain. Oh, you need that stuff to coat around your nerve fibers. No problem. I'll have Charlie over there cook up some stuff for you and send it up. That's all it is they're working with them, because primarily the microbiome is in your guts. That's where all of the raw materials come in. They're the receiving docks. and then these guys break it all up and they send it to wherever it goes. It's bi-directional. You know, highway going north, freeway coming south. All right, once again, this is strictly research, but I looked up, you know, the microbiota implications of COVID-19, and this all came up. Rapid resolution of COVID-19 after fecal microbiota, or whatever that is. We're going to find out in a second. And these are all scholarly articles for rapid resolution of COVID-19 after fecal transplant. Fecal, they call it FMT, fecal microbiota transplant. That, because in your feces is where all these bacteria live. Now they're talking about rapid resolution. I'm going to look into this and make a couple of quick statements, but this is not me. This is not me, and I'm not telling you what to do or not to do. Research. This is National Institute of Health. These are people that are speaking in this domain, and I'm all just talking about what they're saying. Okay, so again, this just popped up. And this is from the National Library of Medicine. Not my words. Not a single word from me is about this. But this popped up, rapid resolution of COVID-19 after a fecal microbiota transplant. All right, and then they're talking about all these different articles that are cited about looking into this, why the gut-lung axis works, and the gut microbiome disruption following this COVID, and developing management gastrointestinal symptoms in long COVID. So there's some work being done here in the research realm, but it was interesting to see that they're claiming some kind of rapid resolution. Well, I don't know. I don't know anything about that. Like I say, I just do research. I am going to be looking into this, but I just figured I would, you know, because Diana is a researcher as well, and if you know Diana, I'd love to have you just put it past her. I'd love to converse with her about this. I know she's desperately ill. When I looked at her X page, it was heartbreaking. She's in bed just crying all day long, and you know, if it's just a matter of putting some bacteria back in her to create the enzymes. And this is the thing, we don't have a bacterial database. We were trying to get one created in North Ireland because they have some terrible problems with autism, and the kids over there are really suffering. And they are everywhere now. Everybody's getting autism, some kind of ADD, or all those, they have terms for everything. However, I see it as a declining intake of bacteria, basically, that's good for you. And, you know, there's so many different things that cause these issues. All the way from the way the mother is you know, her diet and everything else, and her intake, and how they take care of the babies at the hospital. Sometimes they use antibiotics. You know, there's a whole series of things that have to happen in your body to create the probiotic environment that does all the work in you. The probiotics, and that's why there's so many, see everything about probiotics now. Oh, probiotic this, probiotic. I can tell you this right now, and this is just, again, research. From my research, and all the way back to Hippocrates, thousands of years ago, he said all disease starts in the gut. I'm pretty much on it with my friend Hippocrates. I go along with that as well. Gut, brain axis, gut, lung axis. I'm telling you, it's gut, energy axis. They're going to find that everything is related to that. Because if you don't have the enzyme, you're not going to get the product. It's just as simple as that. You don't have the product, that's the chemistry. The chemistry makes everything work. And I originally looked into COVID-19 when it first came out, and it is primarily almost like pneumonia, basically. It's a very similar situation. And I believe it's attacking one of those layers of tissue that is the seal. And it's got to be something to do with the rubbery one, because it's primarily in the lung, although you do breathe in the lung. However, it also affected the heart, as I recall, the rubberiness of the heart. And that's another thing that's doing this all day long. Your lungs are going like this, your heart's going like this. Your lungs are doing this. Now, I'm sure those are, I think primarily those are the two areas where COVID is most injurious. Well, I could design a great study for this. Very, very quick, easy, simple swabs of fecal matter and so forth. Now, SARS-2 COVID increases the inflammatory share of macrophages in the heart, leading to heart damage. And macrophages are supposed to protect you. It says this change appears to result from the immune response to the lung injury rather than from viral infection of the heart itself. So they're looking for a viral infection of the heart. Well, they're not seeing the virus. However, are they looking at the Slurpee? The Slurpee, I think, the gooey stuff is the part that I believe is causing the damage. Not that the actual tissue is being damaged, but once you start not having the adhesion correct, then you're, and because they both do that, they both move a lot. So I would say it's probably one of the lower layers, the second layer down or the third layer down. It's the one that's getting more damage. I wonder if they could actually see that. Okay, here we go again. We may have found a target for treating the fatigue of long COVID. Well, let's see what they have to say. Let's just cut right to the chase. They're talking about interleukin 6. All right. So it says in humans, inflammation causes neurons to release this immune cytokine, interleukin 6. The team found a comparable protein in their test animals traveled to their muscles via the bloodstream and activated a cellular program called JAK-STAT. It turned down the amount of energy produced by the muscle tissues, mitochondria power plants. Now, these are the little glowy parts, mean there's some energy going on. This is what happens at, well, here, let me read it to you. Violet staining of a fruit fly muscle, these are fruit fly muscles, indicates how well mitochondria, which are the power plants, are producing cellular energy in healthy muscle. Left. And muscle affected by pathways resulting from central nervous system stress, which is here. Well, what's central nervous system stress? I believe it's either the enzymes are missing or you have too much of the bad enzymes. These are protected, these little tubes of energy, basically muscles. Muscles are you can see this looks like it has some going on in here, but these are, not too bad. They're glowing pretty good, but this is just nice, like it's doing some work. This just doesn't have any energy to it, because you need to be able to see some brightness. To see, anytime you see brightness, it's energy, whether it's in light or it's in an explosion or whether it's in muscle. Light is energy. Now see, I think they might have this backwards. They say that we're not sure why the brain produces a protein signal that is so damaging to the muscle function across so many different disease categories. I think it's because the coating that is the membrane is affected by all kinds of different types of tissue. That same coating is used in many different areas. And I think it's related to a lot of like this kind of stuff, muscle, cardiac muscle, lungs expanding and so forth. I don't know how much muscle you have actually in your lung. I never really looked at that. You see that? No, your lungs don't have any muscles. This stuff, every day I think of something that I never thought about before. Other muscles in your body help control your breathing, so your chest muscles and all that stuff, but they're not in your lungs. And I never saw any in there, so I kind of suspected there wasn't any, but, I never really gave it much thought. Okay, like I said, just research, but it's kind of interesting. They're talking about long COVID may be caused by chronic cytokine signaling. Well, cytokine are signals sent out to create like a storm, they call it a cytokine storm, which your body turns on itself. Now they say some parts of this puzzle remain unclear. Whoops. All right, as I've been saying, this is all about invasion of the membranes. It says like any other form of acute inflammation, the permeability of the surrounding vasculature, which is the membranes, in this case is that of the lungs, occurs as the lung permeability, all of the little holes, rises. Protein-rich pulmonary edema fluid is drawn into the lungs, ultimately leading to respiratory insufficiency. Protein-rich pulmonary edema. This is, you were open to invasion, and the people that had that open to invasion biology in their body got this particular COVID, and they just never recovered. Is my feeling is, and my opinion as a researcher only is that we should do a bacterial analysis. And actually, maybe somewhere down the line, do some experimental fecal matter transplants, because they did say spontaneous. What was it? Here's right here. Rapid resolution COVID-19 after fecal matter transplant, and that's if you ever have gut distress, digestion problems, a lot of gas and flatulence and smells and all that stuff is supposed to be controlled by bacteria. You're supposed to have be able to process your food, passes through you without any really serious, no constipation, no real diarrhea, you know, a little fluctuation of this and that. Our diets now, we have all kinds of things going on. But if you have like regular diarrhea or regular constipation, then you got a problem. Basically because you're not digesting correctly. It's supposed to be sort of a nice, like Marguerite says, a sausage. All right, I'm going to leave it at that, but I just figured I would address this, and I'll go into the dark matter later. But that's why I just popped right up on her. What makes up 95% of the universe? She says the matter and energy in the universe is 95%. Chew on that for a minute. Well, I hope she's going to get better, and that's just my 2 cents I'm putting out there, because I'm a researcher, she's a researcher. I would love to engage with her, and you know, it's heartbreaking to see her on her page. She's in desperate trouble, and I all I can assume is she doesn't have the chemistry, and if she has any digestive issues, I'm sure I'd be very surprised if she doesn't. Because that's the key. If you're not digesting right, nothing's going to work right. And the worse your digestion is, the worse you are going to be. And some digestion issues affect your neurons, your nervous system. Some affect your other issues like lungs and energy and mitochondria and all that stuff. So there's a lot to research here, but you got to get better first. Let's get you better. I see you got a lot of contacts. You got a lot of contacts. If you just saw what I presented, and I hope somebody can get through to her, because very few people see my stuff. And she's got a lot of contacts that could actually help. Now, fecal matter transplants, I'm just talking off the top of my head now. I'm not saying it's a good thing or a bad thing or anything. But the only way they're using them now is for C. diff, which is a bacterial infection, and they give you a whole bunch of antibiotics that kills all that stuff in you, and then they flush you with new probiotic, fecal matter, and it cures it almost all the time. And basically, that's almost the only way to do it. Now, if it's approved for that, I was told that if something is approved for something, there's something that's called off-label use. So they say, well, we're not going to use it for C. diff, but we're going to use it for something else. Now, I don't know if that's true or not. And again, I'm not a prescriber, I'm not an advisor on none of that stuff. But I'm just saying, research is research. Now, who's doing the research? I can see that there is a lot of research on these fecal matter transplants, very positive, but I don't see it in the mainstream talked about a lot. But I do see people, a lot of people with this long COVID, a lot of people have mentioned it to me. I don't say do anything, don't do anything. I just say go. If you got gut distress, if you can fix your gut, normally that clears up a lot of things. Just as Hippocrates said. All right, this is tragic. I mean, she was vital. She's attractive. She's happy. She's, very engaging, thoughtful, all of the good things. And boom, now here she is. Look at this. Diana is in bed and is not sleeping. She has to close her eyes and rest to avoid crashing. The live stream is not frozen. Diana cannot move much at all, which is a true day in the life of someone with severe COVID. Severe long COVID, or ME/CFS. Now, again, I say everything is due to chemistry. I would like to know what her bacteria is, the different types of bacteria. That's where I would go. But we don't really have a good database yet on that. And again, she has a very, very good following, and a lot of people are very concerned about her, as I am. And this may be a way to get someone to take a look at this. Because again, if you saw what I just presented to you, here's the thing. They didn't even realize this membrane existed. Where is it? I got it around here somewhere. No, maybe not. But there's a membrane that is, hold on. All right, so this is the membrane where all that bacteria and enzymes live, right in here. And this was just discovered in 2018. I don't know whether I showed you this or not. I got so many interrupted so many times. But I would like to see the study done to find out what is the natural membrane in the human body. All right, I'm sorry, my friends, I got interrupted a couple times, but I'm just going to leave it at this. They say they think they found a target for treating the fatigue of long COVID, and they're talking about cytokine storms. I'm saying let's look at the bacteria that creates the enzymes, and I believe it's enzymes. And it is also for Alzheimer's and Parkinson's. They're finding the same thing. Neurodegenerative diseases have similar things. All of them have the same thing, is you don't have the chemistry. You don't have the chemistry, and because you don't have the bacteria. The bacteria create the enzymes are the chemistry. That's why I say, now that we're talking about all of the different pathways in your body are serviced by, like, this is the brain-muscle axis now. All right, and they're relating this to respiratory infections. Well, that could be because the brain and muscle is the same thing. It's one of these gooey things. Respiratory is one of these things. Heart is one of these things. These are the ones that are being invaded, and the cerebral fluids. And certain conditions will manifest because certain enzymes are either in excess or missing. It's basically, that's the best I can do. I would say we have to find out the bacteria that exist in all these people and see if there's a consistent pattern between people that have very long and very severe COVID versus the ones that get over it right away or never got it. And I think we're going to find something there. All right, so, oh boy, boost brain function in older people. I got to read that. All right, there's a lot to know now. There's a lot to think about. So let's keep thinking, all right? I love you all. Take care. Thank you. And give Physics Girl your prayers. And I hope she sees this, or someone can get this to her, that she at least understands what I'm showing. Because I put pieces together. I know that all these pieces are out there. Oh, she should know this, she should know that. Well, maybe. But when you put them all together in a row, sometimes it's a little more understandable. And I think it all boils down to the creator of the chemistry, which is the bacteria creating the ribosome being a little tiny ball floating around through your body, opening up into the enzyme and doing the chemistry. And without the bacteria, nothing happens after that. And bacteria haven't been doing all that well with all the things we've been ingesting and all the toxins and so forth. And bacteria are little living creatures. They live in our fluids. And when you inundate those fluids with all kinds of things, alcohol and nicotine and all kinds of different drugs and this and that, who knows what, and arsenic and water supplies, all kinds of things can affect the bacteria in your body. But to have a database of knowing what bacteria is supposed to be in what body, and you don't have it, well, then we got something to go by. But right now, it's just all guesswork. Anyway, Physics Girl, I love you. I hope you do well. And God bless you for having such a wonderful husband. I've been sort of keeping my eye on you here. I hope things work out for you. All right, thank you.